Okay, so have you ever felt overwhelmed by like a mountain of research? Like you've got this stack of papers on your desk and it feels impossible to get through it all.
Oh, absolutely. Especially with a topic as complex as gliomas, there's always something new to learn.
Right. Well, that's what this deep dive is all about. We're taking a deep dive into the research you provided, sifting through the jargon and pulling out the need-to-know nuggets.
Mhm.
Today, we're zeroing in on adult-type diffuse high-grade gliomas and this fascinating research article. Proteomics separates adult-type diffuse high-grade gliomas in metabolic subgroups independent of 1p19q co-deletion and across IDH mutational status.
Now that's a mouthful.
It is, right. But don't worry, we're going to unpack it all. And you
Okay.
know what's really cool about this research? It kind of flips the script on how we think about genetic markers in tumors.
Yeah, it really challenges some of our assumptions.
It really does. We tend to think of these genetic markers as static, but this research suggests there might be more to the story, especially when it comes to glioma behavior.
Absolutely. But before we get too deep into that, maybe we should do a quick refresher on gliomas in general, just to make sure we're all on the same page.
Yeah, good idea.
So gliomas, as you know, are a type of malignant brain tumor. They account for a significant portion, about 80%, of adult brain cancers. And they can be very challenging to treat.
Yeah, I remember reading that the 5-year survival rate is unfortunately really low. So how does this research offer a new perspective on these tumors?
Well, right now, the way we classify gliomas is primarily based on the World Health Organization grading system. This system considers factors like IDH mutations and 1p19q co-deletion status. It's basically looking for specific genetic changes, kind of like typos in the tumor's DNA.
Okay, so these typos can give us some clues about how the tumor might behave. But this study takes it a step further with proteomics, right? And what I find so fascinating about proteomics is that it's like instead of just looking at the blueprint of the DNA, we're actually seeing the building being constructed. We're seeing the proteins that the tumor is actively creating.
Precisely, and that's a key distinction.
Huh.
While genomics and transcriptomics have provided invaluable insights, proteomics offers a more dynamic and functional understanding. We're not just looking at the genetic code itself, we're looking at the proteins, those workhorses of the cells, carrying out those instructions. And their activity, what they're actually doing, can change, even if the genetic code stays the same.
So it's like getting a more complete picture, a more real-time snapshot of what's happening within the tumor. So what exactly did this research uncover using this proteomics lens?
Well, they discovered that IDH-mutant gliomas, which up until now we've generally grouped together, can actually be divided into two very distinct subtypes based on their protein profiles.
Wait, so even though they have similar genetic, those typos you were talking about, they're showing different behaviors.
Exactly. And these subtypes, they're called HGG IDH-mutant and HGG IDH-mutant. And the really interesting thing is that they were different from each other regardless of their 1p19q co-deletion status.
Okay, now that is interesting. So for a long time, we thought that 1p19q co-deletion status was like a major factor in differentiating these IDH mutant gliomas, and now this research is saying, hold on, not so fast.
That's exactly right. It's like we've been looking at these tumors through one lens, and now proteomics is giving us a whole new perspective.
So it's not just a minor difference, right? We're talking about distinct behaviors. What did they find?
The study found that these IDH-mutant gliomas, despite having those similar genetic markers, were as different from each other in some ways as they were from IDH-wild-type gliomas, especially when it came to their metabolic profiles.
Their metabolic profile, so how they're processing energy, essentially.
Exactly. And that's where things get really fascinating. One of the IDH-mutant glioma subtypes, the HGG IDH-mutant, showed signs of relying heavily on a process called glycolysis for energy.
Glycolysis. Now, isn't that connected to like how some cancer cells seem to crave sugar? They're like sugar addicts?
You got it. It's a hallmark of many aggressive cancers, and it's known as the Warburg effect. These tumor cells gobble up sugar at a much higher rate than normal cells, but they're not very efficient at processing it. It creates this sort of metabolic chaos.
Okay, so metabolic chaos, definitely not good news. But could this insatiable appetite for sugar, could that be a weakness? Could we potentially exploit that?
Now you're thinking like a researcher? That's the million-dollar question, and it's precisely what makes this research so compelling. By identifying these distinct metabolic subtypes, we're essentially uncovering new vulnerabilities within these tumors.
I love it. So I've got these distinct subtypes, different ways they're processing energy. But before we get too far ahead of ourselves, how do we know this wasn't just a one-off finding?
That's a great question, and that's where it gets even more exciting. They didn't just look at one data set. They actually validated their findings across 3 other independent data sets of glioma patients.
Oh, wow. So this wasn't a fluke. They're seeing the same metabolic split across the board.
They did.
Yeah.
And here's the real kicker. They found these subtypes, high oxidative phosphorylation and low oxidative phosphorylation, regardless of the IDH mutation status.
Wait, so even in gliomas that don't have those IDH mutations, they're still seeing these different metabolic
What
profiles.
exactly? And get this, they even saw that these metabolic subtypes seem to align with other molecular subtypes of gliomas, like proneural, classical, and mesenchymal.
Hold on. So you're saying these IDH-mutant tumors, which we thought were their own distinct category, are showing similarities with other types of gliomas that don't even have the IDH mutation based on their metabolism.
You got it. It's like we've uncovered this hidden connection between these tumors, something that goes beyond those initial genetic classifications.
This is incredible. It's like finding a secret passageway between different parts of the glioma world.
I like that. A secret passageway. That's a great way to put it. And it suggests that there might be some fundamental underlying mechanism driving these metabolic differences, something that we haven't fully understood yet.
Okay, so for our listeners who are probably already thinking about their next research presentation, what does this mean for the future of glioma treatment? Are we talking about new therapies here?
Absolutely.
Yeah.
By understanding these metabolic subtypes and how they differ, we can start developing therapies that specifically target those vulnerabilities. Imagine being able to tailor treatment plans from day one, giving patients the best possible chance at beating this disease.
It's like we're moving away from this one-size-fits-all approach to a much more personalized approach to glioma treatment.
Exactly. We're talking about potentially developing drugs that, for example, disrupt the energy production of those sugar-hungry tumor cells.
Mm.
We can cut off their fuel supply, and because we're targeting specific metabolic pathways, we can potentially minimize the side effects on healthy cells.
This is incredibly promising, but like with any good research, I'm sure this raises even more questions, right?
Oh, absolutely. We've learned so much, but there's still a lot we don't know. For example, why do these subtypes exist in the first place? What's driving that? Is it related to how the tumor evolves? Could it be linked to the type of cell that the tumor originated from? These are all questions that researchers are eager to explore further.
It's like opening one door to reveal a whole new hallway of doors, and
Ah.
each one leads to even more questions and possibilities. It's so exciting.
Exactly. And that's what makes this research so groundbreaking. It reminds us that our understanding of cancer is constantly evolving. There's always more to learn, more to explore, and that by challenging our assumptions, we can make incredible discoveries.
This has been an incredible deep dive already. We started with what seemed like a mountain of research on gliomas, and just by digging into this one study, we've uncovered a whole new way of looking at these tumors.
We've seen how proteomics is revealing the hidden world of tumor metabolism, and how these insights could pave the way for more targeted and hopefully much more effective therapies.
And for our listeners out there, you don't have to be a scientist to appreciate the significance of this. This research gives us hope. Hope for a future where glioma treatment is more personalized, more effective, and ultimately more successful.
It really is amazing to think about the possibilities. And you know what's really exciting about this research? It's not just theoretical. It has real potential to change how we treat gliomas in the clinic.
Okay, so let's talk about that. How do we translate these findings from the lab bench to the patient's bedside?
Well, imagine this. A patient comes in, they've just been diagnosed with a glioma. We take a biopsy, and instead of just looking at the genetic markers, we analyze the tumor's protein profile. We figure out, okay, which metabolic subtype does this tumor fall into?
So we're talking about truly personalized treatment here.
Exactly. Knowing the tumor's metabolic type is like having a secret weapon.
So, for example, with those tumors that are hooked on sugar, the ones relying heavily on glycolysis.
Right, the one stuck in that Warburg effect.
Yeah, exactly. We could potentially hit them with drugs that specifically interfere with their sugar metabolism.
It's like cutting off the fuel supply.
Okay.
Starve the tumor cells that are dependent on that sugar rush.
And for the other subtypes, the ones that aren't so reliant on sugar, maybe we explore different treatment avenues, tailor the approach based on their unique metabolic fingerprint.
Precisely. The key here is understanding each tumor's metabolic strategy, its own Achilles heel, and then we can develop these targeted therapies to disrupt it.
This is incredible. It's like we're finally starting to understand these tumors at a whole new level. It makes you wonder, though, what other hidden complexities are we missing when it comes to cancer? What other assumptions do we need to challenge?
You hit the nail on the head, and honestly, that's what keeps me coming back to the lab every single day. It's the excitement of the unknown, the possibility of uncovering those hidden connections that could change everything, and this research is a perfect example of that.
Absolutely. And it's a good reminder for our listeners, too. Never stop questioning, never stop exploring. There's always more to learn. This deep dive has been amazing, by the way. We started with this mountain of research on gliomas, and through this one study, we've uncovered a whole new way of looking at these tumors.
We've challenged old assumptions, we've sparked new questions, and hopefully we've ignited a sense of excitement about the future of glioma research and treatment.
For sure. But like you said, this is just the beginning. So for our listeners who are ready to dive back into that mountain of research, what's one thing you want them to keep in mind? What's one pearl of wisdom you can leave them with?
Don't be afraid to challenge the dogma. Don't be afraid to ask the what-if questions. This research is a perfect example of how challenging those existing paradigms, thinking outside the box, can lead to these groundbreaking discoveries that have the potential to change lives.
It's about pushing those boundaries, right? Looking beyond the surface and asking those tough questions because who knows? The next big breakthrough could come from a question that no one else has dared to ask. It really feels like we've stumbled onto something big here. Like this research wasn't just another study, it was like a turning point.
I agree. It's definitely a shift in how we think about these tumors.
So as we kind of wrap up this deep dive into the world of gliomas and this incredible research on metabolic subtypes, what's the big takeaway for our listeners? What do you really want them to walk away with?
For me, it really boils down to the importance of looking beyond the surface. We could study something like gliomas for years. We can map their genetic makeup. But this research highlights that there's always more to the story.
It's like that saying, the map is not the territory, right?
Exactly. And in this case, the territory is even more complex and fascinating than we initially thought.
We've been looking at these genetic markers, these mutations, as the primary way to understand these tumors. But this research shows us that even within those categories, there's this whole other layer of complexity.
Exactly. And that layer, that complexity, is all about how the tumor is functioning, how it's metabolizing energy. And that's where those metabolic subtypes come in.
It's like we've been looking at the blueprint of the building, but now we're actually stepping inside and seeing how all the systems work, how the electricity flows, how the plumbing is connected.
And by understanding those systems, by identifying those metabolic differences, we open up this whole new world of possibilities for treatment.
That's what's so exciting about this. We're not just talking about incremental progress here. This research has the potential to revolutionize how we approach glioma treatment, making it much more personalized and hopefully much more effective.
And it all starts with challenging our assumptions, asking those tough questions, and never being afraid to dig a little deeper.
This deep dive has been an incredible journey. We've gone from mountains of research to the intricate metabolic worlds within these tumors. We've challenged old assumptions, sparked new questions, and hopefully left our listeners with a sense of hope and excitement for the future of glioma research and treatment.
Absolutely. And to anyone listening who's facing their own mountain of research, my advice is this. Don't be afraid to get lost in the details. Embrace the complexity, because it's often in those unexpected connections that the most groundbreaking discoveries are made.
That's a great note to end on. So to all our deep divers out there, keep exploring, keep questioning, and keep those brains buzzing. We'll catch you on our next deep dive into the fascinating world of cutting-edge research.